Okay, there's the recording. This is July 21st, 2025. All right, everybody. So here we are in week 3. Week 3 is the halfway point in the 5-week semester. So well done. Now you just have to keep going for another couple weeks. This week, we will be having our midterm exams, right? It's very exciting. You have a lab quiz on Wednesday during our regular lab time. So that means that you have the full 3-hour lab period. You know, I'm getting paid for the 3 hours, so I'll be here as long as you need me here, okay? So that's there. Don't forget, it's open notebook, okay? So bring whatever printed resources you have and you would like, but no iPads, no digital resources. Fair enough? Calculator will be helpful. You could probably do all the math on this with a pen and paper, but, you know, double check it with a calculator too. Why not, right? So I suggest bringing your own calculator if you have a calculator that you love and that you're used to. I'll also have very basic calculators available. They look like this, all right? And they're like teaching calculators. They look, yeah. They can do everything you need, but, you know. It feels nicer using a big TI-83s, TI-84s. You spent the money on one of them and probably used it in one math class and use it again right here, right? All right, so lab quiz on Wednesday. The format, right? It's going to be basically the same format as the end of your lab protocol questions, where like, hey, if you had to make a solution with this concentration, how would you do it? Or, hey, here's a standard curve. What's the concentration in it? Well, here's a standard curve, and then you dilute a sample, and you get a concentration of this. What was the concentration before the dilution? It's just checking the core essential skills that I've been trying to emphasize in the lab, right? Not a lot of conceptual memorization stuff, a lot more application, do a little bit of math. Yes, question?
Do you know approximately how many questions?
I think that there are, I think that there are like 12 to 15 questions on there. In the past, people have completed this in as quickly as 40 minutes, and people have taken the whole 3-hour lab time. And people who have taken the full time or the 40 minutes have both gotten, you know, above 90%. So some people, it'll take longer. Some people will be in and out and then go off on the rest of your Wednesday. Solid. Did that answer? Awesome. Any other questions about the lab quiz on Wednesday? Solid. It's worth 50 points, which, if you look at the other assignments, isn't a huge amount of points. But it's meant to prepare you for the lab final, which is going to be worth a full 100 points. This is slow. 100 points, right? Question?
It's a little early, but will that be cumulative?
Yes. Yes, it will. Both the lecture final and the lab final will be cumulative. However, they will focus on material since the midterm. Yep, pretty standard issue structure right there. Okay. Yes, it's the Scantron 882E. There are 20, let's see, it's a 100-point midterm, and I believe that there are 33 multiple choice questions on it. You get the full hour and 50 minutes to complete it. All right, so yeah, Scantron 882E, and you gotta fill those out with a pencil. Some folks, you know, people don't use Scantrons that much anymore. Instead, they do multiple choice exams on Canvas, which, hey, people can do whatever they want. I think that's kind of silly. But then again, who cares? Who cares what I think? Me, and that's enough. Me and my loved ones care about what I think, and that's all I need. To feel seen, safe, and soothed, right? What are those levels of, is there a fourth one when you're talking about like healthy connection? I've been in a lot of therapy lately. Anybody know that fourth? Huh. Anyway, So we've got your midterm exam for lecture coming on Thursday. Now for that one, multiple choice. I can get you more details. I gotta look at the exam again, make some edits to it. But the study guide is there, and I'm not going to deviate from what's on the study guide, those topics. Question, please.
That one you said we should be able to do based off of the lectures,
Yes.
not so much off of the reading.
Yes. I work to make sure that you could do none of the reading and technically still answer all of these midterm exam questions. Now, if you have the textbook, there are practice questions at the end of each chapter, and some of my questions are pretty closely modeled after those. So that's a free pro tip. If you want to get used to the structure of the questions and the language used in them, that's not a bad idea, you know. Cool. Question, please.
Are some of your questions in midterm like how we've been doing for our quizzes that we've been taking?
For the reading quizzes?
Yeah.
They look similar, but they will all cover material that we've covered in the lecture. Does that answer make sense? Yeah. So if you've been doing all those reading quizzes, which I hope you have, because there's a bunch of points there, you know what I mean? Those reading quizzes also give you an idea of what some of the structure of the questions will look like. Yeah. I think I removed all of them that are like, select 2 of these answers, because those give people a lot of trouble. Yeah. Any other questions about the midterm lecture exam? It's not open note. It's not open notebook or anything. Yeah. All right, cool. If there are no further questions, I did just publish an announcement like 5 minutes ago with the same details in it. If you want a refresher, it's in your announcements. All right. I don't want anybody to come in on Wednesday and be like, wait, what's happening? You know, it'd be no good. Okay. So yeah, if there's no further questions, we can go ahead and get right back into our PowerPoint. Okay? We've got a fun lab today as well, where we're doing pigment extraction and paper chromatography. All the stuff's already out of your benches. Bless you. So all thanks to Ashley for taking care of that. It looks like you're all set up for success. There's ground spinach at the side. We'll pull the pigments out of it, separate all the different pigments, and then we will do paper chromatography. That's how we'll separate them, actually. And then we'll take the extracts of those individual pigments back up to the 96 well plate reader and develop our own absorbance spectra for the different pigments. In the same way that they used to originally derive the absorbance spectra for pigments, but saving us a ton of time because we'll use the fancy equipment instead of having to do it in a tiny little spectrophotometer that's all, analog and stuff. So it'll be fun. Okay, enough dallying, enough dillying. Here's summer session week 3. Biology 105, summer session week 3. All right. Ready? Okay. Metabolism, all right? We're talking enzymes, which I know we did in lab on last Wednesday, all right? But we'll talk about metabolism, enzymes, and then that'll move us pretty easily into our discussions of respiration and photosynthesis. But you can't talk metabolism without talking a little bit about energetics, okay? So first of all, metabolism. All it means is it's the sum of all the chemical reactions going on inside of a cell, or if you're talking about an organism's metabolism, within that organism. If you're talking about an organelle's metabolism, it's all the chemical reactions going on within that organelle, right? Metabolism just means I want to know about all the chemical reactions. Cool? Let's get basic energetics covered and out of the way. There will be overlap between terminology here, but each term that I introduce to you, such as catabolic and anabolic, has a distinct meaning. And if you're ever feeling like you don't understand the distinction between, for example, a catabolic reaction and an exergonic reaction, Don't be shy to let me know, and I'll clarify it. So here we go. We're beginning with catabolism and anabolism. All right? Catabolic and anabolic reactions. Anabolic reactions take small things and build large things. All right? So polymerization would be an example of an anabolic reaction. Very good. So when we join any of those monomers of the biological macromolecules up into polymers, joining them up via dehydration synthesis is an anabolic reaction. Whereas breaking them down, depolymerization, right? So if you're breaking down glycogen to release glucose in your body because your blood sugar is low, well, that is a catabolic reaction, where large molecules are broken down into smaller molecules. Now, I like the Jenga tower analogy for catabolic and anabolic reactions, right? Someday I'll bring in Jenga blocks, but let's imagine this theater of the mind, here we are. Let's imagine that I'm assembling a Jenga tower. So young, he's so fast. Whoa, it's so tall. Careful, careful, it's too tall. That uses up energy, all right? I'm taking a bunch of small pieces and I am building something larger, the tower, the Jenga tower. And here it sits before you. It's perfectly square. I'll push the blocks in. Oh, let me get that one. Like that. There we go. Very good. Okay? You got your big tower, right? Now, a catabolic reaction takes that large molecule and breaks it into smaller pieces. So now, the classroom cat, come here. Well, there it is. Now it's on the table. Oh, hello, kitty. Careful, don't get too excited. The cat walks over to the Jenga tower and knocks it over. That releases a ton of energy, okay? The cat had to put a little bit of energy into it to get the tower to start to teeter-totter, and then it falls down. There's energy stored in the Jenga tower. It breaks into a bunch of small, disorganized pieces, all right? It took a little bit of activation energy to make the whole thing break down, because it was pretty stable. I built it so well, all right? But once you destabilize it, it can sort of cascade and break down and release all that energy. So the catabolic reaction is the breakdown of large molecules into small ones. This leads to a release in energy. And last Wednesday, I talked to you about that small energy input that you need, right, to make that catabolic reaction start. That's our activation energy. Remember that? And remember, enzymes lower the activation energy barrier. So imagine I built that Jenga tower on an angled wooden block. So it was already destabilized. Well, now the cat walks by and it just lightly touches it with its tail and the whole thing falls over. It took way less activation energy. Or maybe just the ambient energy of the room, there's some air movement, and that's enough to knock it over because the activation energy barrier has been lowered so drastically. I'm giving you all the concepts at once, and then we'll talk about each one sort of in more detail. Are we comfortable with the cat analogy? It's nice because it also will help you remember catabolic. Cool? All right, here is a catabolic reaction in a motor, all right, in a car. Every textbook uses this analogy. The car takes gasoline and oxygen, carries out combustion, produces carbon dioxide and water. Gasoline is a large, complicated molecule. You produce smaller molecules and you release a bunch of energy. That energy is either harvested for movement or lost to the system as heat. Usually heat is considered as an inefficiency in every chemical reaction, every chemical reaction. In metabolic reactions, there's usually heat released, all right, due to inefficiencies. The energy can't all be harvested for, say, movement here. So some just radiates back into the environment, okay? So this is a pretty straightforward analogy for cellular respiration. Where in the mitochondria, as long as we're talking about eukaryotic organisms, right? Because prokaryotic organisms don't have mitochondria. In the mitochondria, glucose, a high-energy organic molecule, a complex large molecule, is broken down into carbon dioxide. It reacts with oxygen, producing carbon dioxide and water, both lower energy molecules, smaller molecules, all right? It's a catabolic reaction. Some of the energy gets stored in ATP, and then it can be used for cellular work. And due to natural inefficiencies within any reaction, some energy is lost as heat. Cool? When I was a student, I don't know why this resonated with me. When I was a student, but my instructor said, if a car was 100% efficient at harvesting the energy in gasoline, the engine would never get warm. And I was like, huh, that's interesting. It's interesting. Because the reaction of exploding the gasoline isn't the most efficient way of harvesting the energy that's contained within the gasoline. You see what I mean? But anyway. Energy types, you've seen this before. You know the law, like at least you know a couple thermodynamic laws, right? You know energy can neither be created nor destroyed. You know that one, okay? Types of energy. Kinetic energy is energy of motion. All right, the Jenga blocks fall down, energy of motion. Potential energy, okay, is energy stored, and this can be, this can seem too simple, but it's a little more complicated, right? Energy stored in the location or structure of matter and includes chemical energy. What does that mean? Well, the molecules, the high energy organic molecules that we're breaking down in both combustion and respiration, all right? Those have positions within them, right? They have structure. So the structure of matter is potential energy. In the same way that this penguin has a bunch of potential energy because of its position, it can fall down and harvest that as kinetic energy of motion, all right? So you can have the position of atoms within a large molecule and the position of electrons within atoms. That can also be a type of potential energy that can be harvested. So energy just keeps getting switched between different forms. And every time energy changes forms, some of the energy gets lost as heat. All right? So here's the, oh yeah, and so this is why the idea is that eventually everything runs down to a stable state, because every single time energy changes forms, you lose a little bit that just radiates out into the environment. Okay? All right. We'll talk about closed versus open systems, but a very reasonable question that people don't actually usually ask because you know this isn't happening, right? The people will ask, well, oh, on the Earth then, if it's just a system of conversions of energy between potential and kinetic energy, why is there still any living matter? Why is there still any order? Why hasn't everything fallen to a disordered state? But it's because it's an open system where energy is constantly being provided to the system by the sun, all right? I keep using the phrase system, and a lot of people will use that and never explain what it means. I know I've mentioned this before. I'll keep it pretty brief. But systems are just a way that scientists organize sets of behaviors or organize sets of reactions. Sometimes you just want to talk about what's going on in the mitochondrion. So that's your system, okay? And if that system is receiving energy from an outside source, then it's an open system. Sometimes you want to talk about the system of an organism, or the system of an island, or the system of the planet, or the solar system, all right? It's just setting up the constraints, the boundaries, so that I can tell you what I'm actually looking at. And then you don't worry about what's going on outside of the system. Okay? So you're gonna see me talking about systems more and more. There are books published about we need to move away from thinking about things as systems and accept that everything is interconnected. I, as a scientist, would argue that we all understand that the systems are interconnected, but in order to make theories, hypotheses, and rules, all right, we need to simplify and get things down to the system level. Interconnecting all those systems, well, that's for mathematicians and theorists. Question, please. Exactly, you just make a system of systems. That's exactly it. You have the specialists studying the systems that they can fully understand. Then you need some sort of meta specialist who can connect all those different systems. Somebody who has a fundamental understanding of each of those systems. And that's really important. So there is a place for people who don't want to specialize. If you find that you're only interested in about the 300 class level of information, that's good enough. Because if you learn all the different fields up to like a 300 level university class, you can get those specialists to communicate. They'll trust you because you have some understanding of their terminology and what they're studying. All right? It makes them feel safe and comfortable talking to you. All right. And they probably won't patronize you as much. You know, you read like a Wired article with a specialist scientist. If you're in the field and you're like, oh, they hate this reviewer. They think this reporter is dumb because they simplify stuff so much. Anyway, I'm getting off on a tangent, but I'm trying to incite some interest in, opportunities for you if you like sciences. All right, solar energy, boom, comes into the system. This is the planet's external energy source, making it an open system where energy is consistently being provided. Phototrophs or photoautotrophs, all right, take that energy, that solar energy, and convert it into potential energy in high energy organic compounds. The building blocks they use for making these high energy organic compounds include water, and they strip the hydrogen off of the water, which generates oxygen. All right? So these high energy organic compounds, the one that we're going to track is always going to be glucose, because it's the easiest one to keep track of through this cycle, right? Glucose is a carbohydrate. The hydrogen comes from water. When you yank that hydrogen off of water, you generate oxygen. Fair enough? Okay, cool. Chemotrophs, all right, which are, troph, remember, relates to feeding, all right, feeding. So chemotrophs feed on high energy chemicals, high energy organic compounds, break them down and acquire that energy, convert it into ATP, do cellular work with it, all right. This breakdown releases carbon dioxide and water. We're not going to really talk about the nitrogen cycle. So this nitrate piece, I like it being there because you know how important nitrogen is. I emphasized it when we talked about polypeptides and how, you have an entire sense of taste for detecting nitrogen, essentially. You need nitrogen for making amino acids, all right? The nitrogen cycle is incredibly, incredibly, incredibly important. Hopefully you will talk about that if you take a Botany 105 course or an ecology course here, or chemistry course. If you take bacteriology at the 400 level, then you get to learn about really what goes on with nitrogen, which is cuckoo. It's cuckoo crazy. In the soil, the nitrogen's doing all sorts of stuff, all right? And you know what? You can make an entire career out of understanding the nitrogen cycle in the soil, and that would be a valuable career to humanity, because managing the nitrogen cycle within the soil, all right, nitrogen turnover can affect crop yields massively, and it can be done just by understanding, Oh, we need to control this type of bacteria. We need to encourage this type of bacteria. What type of environment do they like? Let's do a test where we see if we can encourage this strain of bacteria. Oh, we do it. It takes 2 years. That's 2 years. You publish. You get your results out there. Agricultural industry starts to take notice of your management recommendations. They change management. They're able to increase productivity without utilizing chemical pesticides and herbicides and fertilizers. That limits eutrophication of the waterways. Like, there you go. You just had a positive impact on the entire planet, all right, by studying bacteriology. Anyway, if you're interested in bacteriology, please take advantage next year of Mark Wilson, because he is retiring. It is public, so I don't feel bad saying this, but he's a celebrated bacteriologist, okay? So he might call himself a geneticist, a genetics specialist. I don't know. The thing is, he's going to be here next year, and if you're interested in bacteria, get in there now. Well, he's still around before he goes off and lives a glorious life of retirement. Okay. We're completely off track. I'm just excited to see you all again. It's been 4 days. You know what I mean? So the carbon dioxide, that water, that's built up by the phototrophs, the photoautotrophs, into these high-energy organic compounds once again. Cool? All right. One last thing. This says phototrophs, and I really like, there's a few reasons I love this figure, because it doesn't oversimplify for you, okay? I talked, about autotrophs and heterotrophs. Autotrophs make their own food. Heterotrophs have to eat something else to get their food, okay? But phototroph and chemotroph is way more accurate, and it opens things up for all the different varieties of lifestyles that exist out there. There are organisms that are autotrophs that don't photosynthesize, all right? They use, say, thermal energy, like a thermotroph or a thermoautotroph, all right? They feed the entire food chain near thermal vents at the bottom of the ocean where light doesn't reach, right? So if I define autotroph for you as an organism that photosynthesizes, I feel like I'm doing you kind of a disservice. Any questions about this? Remember, every time energy gets changed between forms, some heat gets radiated due to inefficiencies. Cool? Laws of thermodynamics. Here's our important ones. Energy can change form, but it cannot be created or destroyed. That's the big one. And then every time there's an energy transfer or an energy transformation, some energy gets lost into the system, okay? And then that energy can leave the system, all right? This results in an increase of entropy, which is a measure of disorder, all right? Things are naturally trending towards high entropy, disordered states. But by putting energy into the system, you can increase order, okay, and combat entropy. Your textbook uses the example of, as a student, your dorm room's messy, right? You gotta constantly put energy in to keep it in order. Your dorm room's constantly going, falling towards a state of high entropy. I don't know. It's fine, but it's not a great analogy because it's not like that spontaneously happens. You know what I'm saying? If you left your room locked and walked away, everything wouldn't fall onto the floor and stuff. Anyway. Here is a bear. The bear eats a fish. The fish has high energy organic molecules in it. The bear breathes out carbon dioxide and water as it breaks down high energy organic molecules to generate kinetic energy of movement. That energy transformation radiates heat, increasing entropy in the system. Questions? Entropy is a measure of disorder, okay? Try not to get too caught up on this. Gases have higher entropy than liquids, and liquids have higher entropy than solids. Because if you think about it too long, it'll it'll start to freak your bean, man. You don't want your bean freaked. It's no good. All right. Just go with the surface level definition for this. It'll be more effective as you're trying to understand the concepts we discuss at 100 level classes. Does that make sense? Cool, cool. If you get into physics, you'll learn more about this. Sound good? Okay, great. So entropy, disorder. High entropy level, high level of disorder. You put energy in, you can reduce the entropy level. Okay, we're not going to talk about the equations for calculating entropy. We're not going to talk about enthalpy, which is another aspect of entropy that relates more to heat specifically. We're not worried about it. I'm not worried about it. Y'all watching that show? It's a great show. I'm 2 years late for the reference. That's fine. Free energy. Gibbs free energy. All right, this is how we are going to be discussing energy. Free energy is energy that can do work. Don't worry about this part. When temperature and pressure are uniform. You know temperature and pressure are metrics of energy, kind of, because we talked about osmosis, and we talked about how increasing temperature makes things move around more. They have more kinetic energy. Don't worry about that stuff, all right? We're going to talk about Gibbs free energy change here. This symbol's the delta symbol, all right? Delta G means change in energy. If you take notes, right, which is awesome to do, incorporating the delta symbol into your note taking can really help you out, because sometimes you're talking about the change in something, all right? And so you can use a little triangle as a shortcut. It's called the delta because the river delta is an area of change, all right? See, it's a lot of change when the sediment comes down, the nutrients get flushed in. If you have a Mac computer and you hold Alt and press J, that makes a delta symbol. There's a band that used to be cool back when I was cool, all right, called Alt J. They're called Alt J because on a Mac, it makes the delta symbol, which is a symbol for change. Isn't that cool? And they have like a hand thing they do. I think it's like this or something. It's a good band. Check out Alt J. That first album was pretty good. I think they went downhill drastically. But then again, who cares? Who am I? Who am I? Vulture? Does Vulture still exist? All right. Free energy changes. What is it? How do we use it? It describes reactions. It basically tells us, does a reaction release energy or does a reaction require energy? Or you could say, is the reaction catabolic or anabolic? But more accurately, you would say, is the reaction exergonic, releasing energy, or endergonic, requiring energy? All right. Exergonic reactions are a nice big umbrella term. It just means that the reaction releases energy. That way, if you're not sure if a reaction is actually catabolic, like if you're not sure if it's actually breaking a large polymer down into monomers, but you do know it releases energy, you can save yourself any potential embarrassment and just say, oh yeah, it's an exergonic reaction. Oh, that reaction required energy? It's an endergonic reaction. And then if someone's like, oh yeah, that's right, it's anabolic, you'll be like, yep, that's right, I knew that from the very start. No problem. And that's how you just coast right through the cocktail party. You just use the general terms, all right? Endergonic reactions require energy. If a reaction requires energy to build something, and energy cannot be created or destroyed, well, then that thing that got built must now hold some of the energy. Fair enough? Sick. All right. So I'm gonna do one of my famous chalkboard summaries. Everybody loves them. They're famous. All right. We've got exergonic reactions. We've got endergonic reactions. Okay? Exergonic reactions release energy. Endergonic actions require energy. All right. Catabolic reactions are exergonic. Anabolic reactions are endergonic. Cool? And we're going to talk about the relevance of delta G in just a second to continue filling out the table. Okay. So, you've got a reaction, right? Think back, this is, in a normal semester, this would be like, think back a month and a half ago. But for us, it's like, think back a week and a half. Think back a week, okay? And we did talk about reactants versus products, okay? And how some reactions are reversible and all that good stuff. So here we've got a figure. The figure shows the potential energy of molecules on the y-axis. So what this is showing you is you've got a reaction. It's going to go in this direction from reactants to products, because that's what reactions do. They take reactants and they go to products. Okay. In this example, the reactants are high energy. The products are lower energy. Because energy can't be created or destroyed, some energy is going to be released. Maybe it's all lost as heat and is never effectively harvested. Okay? Or maybe it's harvested to drive ATP generation. Maybe you did an exergonic reaction without really being concerned in harvesting any energy. You just did it to get the low energy products out of here. Okay? But the reaction itself went from high energy reactants to low energy products. High energy reactants to low energy products. Cool? The difference in free energy, in this case it's listed as potential energy, but the difference in free energy in G values between the reactants and the products is the amount of energy released. That doesn't mean it's the amount of energy utilized for something useful. It just means this much energy got released. We cool with all that? All right. Delta G equals the energy of the products minus the energy of the reactants. Okay? This number, let's just say that this has 1 Gibbs free energy unit. And this has 3 Gibbs free energy units. We're not even going to talk about the units. I think it's joules. All right, yeah? For Gibbs free energy. Nice, dude. Fuck yeah. Sometimes, you know. I think I lost it every once in a while. And then something like that happens. All right, products, reactants. Check it out. Let's say this had 1 megajoule. And this has 3 megajoules. All right? So when we take energy of the products and subtract energy of the reactants, we're going to get -2 megajoules. What am I trying to get at here? In an exergonic reaction, the delta G value is negative. It is less than 0. Oh wait. Yeah, I did it right. Sorry. In an endergonic reaction, the delta G value is greater than 0. It's a positive number. All right? In an exergonic reaction, delta G equals amount of energy released. Whereas in an endergonic reaction, the delta G equals the amount of energy required. You ever see, there's like people who were really good with chalkboard writing, and they had those long sticks with the piece of chalk at the end, and they could stand like this and then write. It's amazing. It's incredible. I'm really jealous of that skill set because I have to turn my back to you and then cover what I'm writing, which kind of kills the flow, you know? So apologies for killing the flow. You know that I care about the flow. All right. Here's an exergonic reaction. Delta G's right there. All right. Here's an endergonic reaction. Delta G's right there. Any questions? Okay. Very good. Now remember, there are inefficiencies in all of these. So even though this is the amount of energy technically required, it might take more energy than that, okay, due to inefficiencies. And even though this is the amount of energy released, you might get less energy than that due to inefficiencies. Cool, these are two big slides, and this is a big table, you know. I mean big in that it's got a lot of core information about how biologists tend to discuss energetics. Okay, there's that. There's that. All right. ATP. We're only going to talk about it a little bit. It's sort of the short-term energy currency of the cell. All right? ATP looks like this. Adenosine triphosphate. All right? If you really have been like, what they call it, mimetic memory. What's that when you have a photographic memory? There's a name for it. It's not mimetic. Eidetic. Thank you. Points. You're getting points like every day. That's ridiculous. No, it's great. They expire at the end of every day. I'm so sorry. You should have been using them. All right. So if you've got a eidetic? Eidetic. If you've got an eidetic memory, you may see this and think, oh, that looks incredibly familiar. That looks like a nucleotide. And it basically is. ATP, adenosine, remember ATCG for our nucleotides? That's the same A, all right? But this one has 3 phosphate groups attached to the end of it. I told you phosphate groups would be important for energy storage. Here's the sort of the cycle here. You got this ATP molecule. Usually it got generated by harvesting the energy from an exergonic reaction. Then you get some ATP. This ATP, the terminal phosphate group, can be cleaved off of it, and that releases a good amount of energy. All right? So you start with glycogen. You do a catabolic reaction there to get the glucose. You take the glucose, you break it down, and through a complicated process, you end up with some ATP. The glycogen was very stable. The glucose was a little less stable. The ATP is a little less stable, but this ATP can now be used to do cellular work pretty directly. ATP is sort of an all-purpose energy source within the cell. Cool? You can use it to pump things across. You can use it to move a motor protein. You can use it to make a flagellum whip. All right? You can do all sorts of things with ATP. Solid. So the terminal phosphate group gets cleaved. It releases what we call an inorganic phosphate because it now no longer has carbon in it. All right? So it's not an organic molecule if there's no carbon in it. And it releases some energy. Here it was like an organic phosphate because it's attached to these, this sugar and this nitrogenous base, both of which are carbon-based organic molecules. Solid? Okay. So, this looks like a scary figure. Trust me, it's not so bad. What this is showing you is energetically how ATP hydrolysis, which is this exergonic reaction right here, how ATP hydrolysis gets coupled to endergonic reactions, all right, in order to drive those reactions, providing the energy they need. So let's look right here. In this example, we have we have an anabolic endergonic reaction where glutamic acid and ammonia are combined to generate glutamine. Cool? Check out that delta G value. It's positive. Takes 3.4 kilocalories, all right, for every mole of glutamine that gets generated. All right? Cool. Now, let's actually kind of look down here. ATP hydrolysis, this exergonic catabolic reaction, has a negative delta G value of 7.3 kilocalories per mole. That means that if you hydrolyze a mole of ATP, you release more energy than is required to generate a mole of glutamine. And maybe that's tingling something in your brain about why talking about moles is so useful, right? Because we're talking about the same number of particles now, even though they're very different molecules. Maybe the brain tingle didn't happen, and that's okay. That's okay. All right? So check this out. Glutamic acid to generate glutamine, what happens in the system is first you hydrolyze the ATP, you slam that now inorganic phosphate group onto glutamic acid, and then that sort of temporarily stores the energy on the glutamic acid, okay? Which means that now it's at a high energy state, and this glutamine can be at a lower energy state, so this reaction's encouraged. It's a little weird to think about how the phosphate group actually gets used, but phosphorylated intermediates are super common when you're talking about ATP hydrolysis and energy. I don't want you to really worry about that part. I want you to worry more about this. Coupled reaction, okay? There's an endergonic reaction going on here. There's an exergonic reaction going on here. If you take the delta G values for the two reactions and you add them together, you still end up with a negative delta G value. Meaning that it is a net exergonic, bless you, it is a net exergonic reaction, okay? Reactions with negative delta G values, net negative delta G values, sort of can proceed because it means that you're releasing more energy in this reaction than is being consumed. And you know, just one sec, and you know that some of that energy is going to be lost as heat due to inefficiencies. So you need to couple exergonic reactions with endergonic reactions so that you're releasing more energy than the endergonic reaction requires. Question? I did it? Nice. All right, cool. There it is. Okay, so coupled reactions, super common. And if you continue on into, high upper division, like in biochemistry courses, they'll have a number of reactions like this and be like, how much ATP needs to be hydrolyzed in order to make this much glutamine, for example. But I want you to just get the understanding of a coupled reaction and what I mean there. You couple an exergonic to an endergonic. The exergonic provides the energy for the endergonic. And the whole thing's usually catalyzed by an enzyme, right? A big protein blob that pushes all these things into proximity with one another. To keep it really simple. What can ATP do? ATP can do so many things. It can do chemical work, where you take that phosphate group, you jam it onto a molecule to make a phosphorylated intermediate, and now that higher energy molecule's reactive, and there might be enough to make it spontaneously react with something else. Okay? Boom, boom, boom. Reactants to products. It can do transport work, all right, where you pump things, okay, across phospholipid bilayer. It can do mechanical work, where you're actually mechanically moving something, like with a motor protein. You blast the motor protein with some energy from ATP hydrolysis, and it goes erk. You blast it again, and it goes erk, like that. So it's mechanical work. Or you're attached to a muscle fiber, and you blast it with ATP, and it goes erk, like that. And it goes zap, zap, zap. And that lets you pull on those muscle fibers, right? Which should be true fibers, right? Microfilaments or intermediate filaments, because you know that microtubules don't do great with tolerating tension, right? They're better at tolerating pressure. We had a whole talk about it. You all loved it. Continuing, okay, ATP's the short-term energy currency for the cell. We cool with that? Once you convert the stuff into ATP, you can use it to do all sorts of things inside the cell. So here's a cycle we've got, okay? Cellular respiration happens, and this is, cellular respiration happens, it generates ATP using energy from other molecules, okay? Then you hydrolyze ATP, and that releases energy. That energy can be used for cellular work. This generates a DP and an inorganic phosphate group, which then can be joined back together with an energy input. So you've got here essentially an anabolic reaction and a catabolic reaction, or an endergonic reaction and an exergonic reaction, okay? And it's being driven by the process of cellular respiration. Cellular respiration is what your mitochondria do, all right? In a prokaryotic organism, it's carried out across the organism's outer membrane in a very similar way. So we'll focus on the eukaryotic story, but I'll touch on the prokaryotic story briefly because the mitochondria had their origins as free-living bacteria, right? So they had to be capable of cellular respiration without being mitochondria. That's why they were valuable. Anyway, enzymes. I drew this on the board last week during Wednesday. We got a little, sometimes in summer school, we get a little bit ahead of lecture in lab or a little bit ahead of lab with lecture. And that's just a consequence of compressing an entire semester down, you know? But let's look here at sort of this is the same framework, the same figure as the figures that I put up before to explain endergonic versus exergonic reactions. Now, you just kind of put time on the x-axis, and you're watching the reactions go as a line, all right? Make sense? Let's take a look here. Sorry, I got a little, let's look at this picture while I grab a paper towel, because I got like a thing going on here. It's called a booger. You know what it is. I don't need to sugarcoat it for you. All right, I hope you enjoyed that, because I'm wearing a microphone. For anybody watching the recording, because I have a microphone basically pressed right up to my nose. Great. All right. I feel like I didn't get it all. 1 second. Good stuff, good stuff. Sorry, I thought I could make it to the 5-minute break, which we're almost at, but I want to get through this figure right here. Ready? Okay. We're good. Reactants here, products here. You're going from high energy reactants to low energy products. What type of, if this is the direction of the reaction, what type of reaction is this? Exergonic, flawless. What other type of reaction would this be? Catabolic. Or whatever other type of reaction could this be, I mean. What's the delta G value here? Negative. Yeah, perfect. It's all right there. I'm just bringing it all back. And this reaction will be releasing energy. Okay? So check this out. You already know on this slide that C represents the delta G value. But what's going on here? Okay? What's this A and this B portion? What's going on with this line? Why does it go up? Here's the idea, right? I already laid the groundwork for this, talking about the cat knocking over the Jenga tower. You have to put a little energy into the reaction to get it to happen. Think about a block of wood, all right? A piece of kindling, okay? Piece of kindling sitting right here. I take it, I hit it on the table, I'm putting energy into it, but it doesn't burst into flames, right? You know that piece of kindling has a bunch of energy in it in all of the structure that its organic, that its molecules have, okay? If you put enough energy in by, say, burning some paper, okay, or just hitting it with some gas and like letting it go, right? If you put enough energy in, you can get the exergonic reactions to start. Then it sort of cascades, right? Where molecule breaks down, releases enough energy to break another molecule down. So a piece of kindling has a bunch of energy in it. It's a high energy reactant, but you have to put something else in there to get the bonds to start breaking. Something needs to destabilize it. All right? Cool so far? Same thing if we're talking about small scale chemical reactions, right? High energy reactants going to low energy products. This reaction wants to happen, in big quotes, okay? But the reactants are stable. Otherwise, we'd all burst into flames, you know? But all of our molecules are relatively stable, okay? Even though there's a lot of energy in them. Solid? So what a catalyst does in general is it reduces the amount of energy it takes for a reaction to occur. A catalyst encourages reactions to occur, but a catalyst cannot on its own make those reactions occur. There still needs to be some initial input of energy. The initial input of energy is what we call activation energy, and it's represented right here. So a normal exergonic reaction like this might have an activation energy represented by A. If an enzyme is present, the activation energy barrier could be reduced and could be B. So A is representing the activation energy for this reaction without an enzyme, whereas B is representing the activation energy for this reaction in the presence of an enzyme. It's going to make the reaction more likely to happen. And I'll give you the last analogy, which worked when I was a student for me, is imagine that this is your house, and this is the park, and the park is downhill. And your friends say, let's go to the park, ride your bicycle here. Now here's the problem. You got a big hill between you and the park. Most of the time, you ain't gonna make it, all right? You're gonna be like, yeah, there's that big hill there, I'm not gonna do it unless I can get a ride, all right? But if some construction project came by and reduced the height of that hill, then maybe you're more likely to do that reaction, to go down to the park, okay? Now technically, when you ride up this hill, you put energy in and you get it back. You store the energy in position, in potential energy of going to the top of this hill. And then you do get it back in kinetic energy here. But it doesn't feel like that, really. Maybe you go wee down the hill, and I think that's exciting, okay? But you lose some energy due to inefficiencies, blah, 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 all that stuff, right? Now, this lower barrier to activation energy, you know, makes it more likely that the exergonic reaction can proceed. And in a chemical reaction, sometimes an enzyme lowers the activation energy enough that just the ambient energy of the system, right, the energy of the molecules wiggling around, might be enough for the exergonic reaction to proceed in sort of a spontaneous way. Question, please.
What is C?
C is the delta G value, the amount of energy released. All right? And it's very important that enzymes do not affect C, okay? Enzymes don't change how much energy the reactants had, or how much energy the products have. But enzymes do change the activation energy and encourage the reaction to proceed. Reducing, I know I'm rambling on this slide way too long, but let me just catch you with one last thing and then it's 5-minute break, okay? Check this out, right? Reducing that activation energy barrier, you always get the activation energy back, all right? Depending on how good you are at harvesting it, that's a wholly different question, right? So it doesn't change how much energy gets released by the reaction. It just changes how much energy it takes to start the reaction off. Are we cool with all that? Okay. Our next slide will be about enzymes, active sites, substrates. This basically moves us away from our energetics talk entirely into enzyme/protein structure. So I'm going to start a 5-minute timer right now. Pause this. Okay, recording resumes. Wait, can you not? Okay, good, we're good. I just checked during the little 5-minute break, everybody, on the midterm, and it is, like I said, 33 multiple choice questions. All right, so it's 100 points, 33, 3 points each, 1 point for putting your name on the Scantron. Cool? Yeah, and do it because there have been people who don't get that 1 point, and I have to track them down, like, is this yours? And I'm like, no, you don't get that 1 point. So put your name on there. Why not? Cool. During the break, did anybody get additional questions that popped in your mind about anything we've talked about so far, or exams, or whatever?
Just on the study guide.
Yeah.
One of the questions was about like pH. What's the change in hydrogen ions? Like a change of 1 pH. I didn't quite understand how to answer that.
Oh, yeah. That's great. Let me go ahead and I'll answer that right now. Did everybody get this table down? Who wants this table? It's a good table. You can take a picture if you want. I'll wait to erase it. Thank you for the compliment on my table. I like this table. All right, I'm going to get rid of it. Here we go. So you got that question there, which was, from the study guide, a change in pH of 1. What does it actually represent in changes of hydrogen ion concentrations? I've repeated it correctly? Sick. Okay, let's just show 3 pHs. A pH of 1, a pH of 2, and a pH of 3. All right, so a pH of 1 means the concentration of hydrogen ions equals 1 times 10 to the negative 1 molar. A pH of 2 would be 1 times 10 to the negative 2 molar. And a pH of 3 is 1 times 10 to the negative 3 molar. Okay? Cool, cool, cool. I could organize this as a table too, but too late now. All right? So now let's actually convert that into a decimal, and you'll see pretty clearly what I'm trying to get at. This is 0.1 molar, this is 0.01 molar, and this is 0.001 molar. So a change of 1 in pH is a tenfold change, or this is 10 times more. pH 1 is a 10 times higher concentration of hydrogen ions than pH 2. Fair enough? That's what I'm trying to get at, just that straightforward. Yep. If we change the pH by 3, how much would it be to do, you know? Cool. How much would it be to do? Good stuff. I've got I'm carrying my extra key to my back gate in my pocket, which is no good. It lives in there sometimes, yes. All right, cool. Ready? Sick. And if you have any questions like that popped up about the study guide, if you're looking at the study guide and you come in and you got a question, don't be shy, ask me. I love answering study guide questions. It's fun. Enzymes. Let's move away from the energetics talk and into the structure talk of enzymes. So, what do we know about enzymes, people, already? They're catalysts. That's solid. Let's go make that 1 point. You get a point for that one. Absolutely, yeah. They decrease the activation energy required. That's the point. Now, push your minds back into the distant past of weeks 1 and 2. Yes. They are proteins. That's worth 2 points. I'm sorry. All right. So, what's the last fun thing we know about them as far as how we name them? ACE. There you go. That's also worth 2 points. Very good. So, they end in ASE like lactase or cellulase or amylase. All right? They are proteins. They are catalysts. They lower activation energy barriers, is how I'd say it. All right? Let's talk a little bit more about enzymes. All right? Enzymes bind things. This red thing is being bound by the blue thing. The blue thing is the enzyme. All right? Enzymes bind things. Those things are called substrate molecules. Okay? So, in other words, an enzyme binds a substrate. All right? The location at which an enzyme binds its substrate is called the active site. Okay? The active site right here, okay, is where you bind the substrate. This forms a complex called an enzyme substrate complex, all right? And somehow that active site reduces the activation energy it takes for a reaction to proceed. Fair enough? So how can it do this? Well, it could orient 2 different substrates correctly. 2 things that otherwise would react, but they always just kind of bounce against each other. It goes, no, listen, let's get you both here and push you together, and now you can react, okay? It can strain substrate bonds. So if you've got a molecule that is made up of both of my forearms, and it has a weak point here, well, it binds into the active site, and it's physically strained, and then it just goes pop. That's a catabolic reaction. That's exergonic. It's energetically favored. I haven't used the word spontaneous yet, because it's an obnoxious word, because it's used so much in our common parlance, right? That then switching to, anyways, here's the point, right? Reactions that release energy, exergonic reactions, they're often referred to as spontaneous reactions because they can technically proceed spontaneously. Do you see what I mean? However, that doesn't mean that they proceed spontaneously because of activation energy barriers. So you don't have to worry a ton about spontaneous. Just know that in chemistry, it means different things than in life. Yeah. Okay. How else? Providing a favorable microenvironment. Remember when we talked about our different amino acids and their side chains, and I showed you how some of them are straight up acidic or straight up basic? Well, this active site right here might be a little pocket of acidity. And when the thing goes in there, well, this change in concentration of hydrogen ions inside of that environment causes the exergonic reaction to proceed more actively. All right? It can also straight up bond to the substrate. If you covalently bond to something that is covalently bond, let's say that, look this, right? These 2 things are covalently bonded, my palm, my fists. They're covalently bonded to each other. And now I'm the enzyme, and I go, covalently bond here. And now I just broke it up, right? Because I've taken up that covalent bond. Make sense? Of course. Biochemistry. You'll learn more about it. So here's an example with, you've got these reactants here, molecule A, B, molecule C, D. The products are products A, C, and BD. All right? That means that you have to break these bonds between A and B and C and D. Even though overall the reaction is energetically favored to go and produce these lower energy products, because it's an exergonic, spontaneous reaction, right? First, you have to put enough energy in to weaken these bonds between A, B, and C, D. Sometimes I I find that this figure is helpful for people. Sometimes I find that it's more confusing for people. All right? So an enzyme can, say strain these bonds directly. See what I'm trying to say? Cool. Okay, cool. An enzyme and its substrate. Very good. Enzyme, substrate, boom, enzyme substrate complex. The enzyme cannot do what it's supposed to do if it can't bind its substrate. Or more specifically, if anything happens to this active site, the enzyme becomes inactive. I used to have this big analogy about lions and gazelles that I made up that I was really proud of, and it was like the active site is the lion's mouth, and the gazelle is the substrate, and the product is its waste. And if, let's say that somebody you came up and you splashed acid into the lion's mouth, well, now it's way worse at using its active site. That's why I don't really use it anymore. It's sad imagining that, okay? Or, here's something more fun. Imagine you find that lion and you give him a big old piece of bubble gum, all right? And now it's got bubble gum all stuck in its active site. That would also reduce the lion's rate of turning gazelles into feces. Okay? I don't know. Sometimes I can't let analogies go. So that's going to be really important as we talk about enzyme regulation. The first example with the acid is a pretty clear-cut example of denaturation of the protein. If the enzyme denatures to the point that its active site doesn't work anymore, the enzyme doesn't work anymore. If you jam something else into the active site and it just sits there, the enzyme doesn't work anymore. Cool? Very good. Physically strains the bond. Boom. Enzyme. Substrate is sucrose. This enzyme is sucrase. The substrate is sucrose. Sucrose ends in OSE. Therefore, you know it is a polysaccharide. It's a carbohydrate. It's a sugar. Boom. You know all this stuff based on the names, all right? Active site. Physically strains the bond. Allows hydrolysis to occur, all right? This is a hydrolytic enzyme in that it encourages a hydrolytic reaction. Releases the products. Now the products don't have an affinity for the active site. The enzyme is now free with its active site exposed again to bind another molecule of sucrose. The idea of this figure is that the enzyme does not get consumed in these enzyme-catalyzed reactions. That's one of those key learning objectives that I have to make sure you all understand about enzymes. The enzyme does not get consumed. Okay? Now the enzyme will wear out like any molecule. Eventually it wears out, needs to be replaced. All right? Awesome. Enzymes have different optimal temperatures and different optimal pHs. So this is an enzyme that does like the typical human enzyme. Has an optimal temperature of 37 degrees Celsius. Makes sense. That's how we live. That's where we're living. So you want the enzymes to be designed to be stable at your body temperature. Heat-loving, a thermophilic bacteria right here, 75 degrees Celsius, all right? This is, I'm hoping just like it's a good thing if in Biology 105, at least the way that I try to teach it, if you're going like, that's okay, okay? That means that it's supposed to all feel simple, you know what I mean? So this is one of those where you could be like, why does this need to be on a slide? Obviously, an organism that lives in a colder environment or has a colder body would have enzymes that have an optimal temperature close to the body temperature or the ambient temperature. Same thing for an organism that lives in a hotter temperature, right? How do you accomplish that? By changes to the sequence of amino acids, by changes to the stability of the molecule. All right? Now imagine this active site a little bit. A protein's a little flexible. So you don't want the whole enzyme to like harden up, just like with a phospholipid bilayer. You don't want it to harden up and freeze. You also don't want it to become too fluid and fall apart and lose its structure and denature, okay? So enzymes are designed for the environments in which they naturally occur. Same thing with the pH. Here's two different enzymes, two different human enzymes, one in the stomach, one in the intestine. The stomach is highly acidic. The intestine is closer to neutral pH, all right? So the enzymes, they're suited to those environments. Straightforward enough? In our enzyme lab last week, if you'd done the 2-day version, you would have exposed enzymes, the same enzyme, the polyphenol oxidase, to different temperatures and to different pHs in order to determine its ideal temperature and pH, right? Good deal. Enzymes. Enzymes determine, to a large extent, a cell's metabolism, okay? The reactions that occur within a cell are determined, to a large extent, by which enzymes are present. This is huge in differences between cell types, okay? So different cell types are different because different chemical reactions are going on inside of them. Fair statement? Intuitive? Very good. Which chemical reactions occur inside of a cell is determined by which enzymes are present, to a large extent. So enzymes, bless you, to a large extent, are what differ between different cell types. We've talked about one other thing, and that's like membrane proteins. That's huge, right? Different membrane proteins can determine different cell functions too. But think about this. It's all proteins, okay? The fundamental unit of the cell is a bunch of sacs of phospholipid bilayers. And then all of these proteins determine what moves between the different sacs and what reactions are occurring within each of the compartments, the sacs, okay? This means that enzymes must be tightly regulated within the cell. So now we're talking about things that affect enzymes, and we're going to use this to talk about enzyme regulation, okay? Sometimes you want a bunch of a certain amino acid, and you've got the enzymes for making that amino acid. So you turn those on, you activate them, you make the amino acid. And now you've got enough of the amino acid. So stop. You got to turn those enzymes off, though, because remember, enzymes don't get consumed in carrying out these reactions. It's cyclical, exactly. So you need to have ways of inactivating the enzymes, and it would be great if you could inactivate the enzyme without destroying the enzyme, because then you can use it again later. So if we're regulating the activity of an enzyme, all we really have to do is affect its active site. Anyways, here we are back to the big text-heavy slide. Factors that affect enzymes. Temperature, pH, the broad chemicals bullet point, which is so broad as to be useless, okay? Sometimes chemicals break up proteins. Fair? Cool. You know about temperature and pH and how those can cause the quaternary, tertiary, down to secondary and primary structure of a polypeptide to unfold, all right, and denature. So you know about temperature and pH here. There are also these are listed as biological factors. The categorization is a little bit wonky, but you'll understand as we talk about this. Sometimes an enzyme needs something that is not part of its peptide sequence, its amino acid sequence, to carry out its function, okay? We'll call these cofactors, okay? It's something else that you need. Oftentimes, it's a charged particle, like an ion, like a magnesium ion, or an iron, an iron ion. All right, it's a charged particle. Because you put charged particles in your active site, that makes the active site more reactive. It could allow a covalent bond to form, for example. It could let you, say, hold oxygen, like in the example of iron in heme, which is in hemoglobin, which is carrying oxygen in your red blood cells as we speak. Look at it go. Feel it coursing through your veins. Oxygen, we know it, we love it. All right. Cofactors, okay? They jam into the active site of the enzyme and they're necessary for the enzyme to function. So, if you want an enzyme to stop functioning, get rid of the cofactors. That's one way of doing it. In the lab, that's done all the time, where it's called chelating. You chelate an essential cofactor in say a test tube, right? You make it all group up and become unavailable. You bind it all up so it can't be in the enzymes. And that prevents the enzymes from operating very effectively. Okay, just as an example. We'll talk about this when we do DNA extractions, I think this week, in our lab. Next, inhibitors, okay? Inhibitors affect enzyme activity, okay? Now, all inhibitors function by affecting the shape of the active site. And these essential cofactors work by getting into the active site. It's all about the active site. Are we cool with that? Awesome. I love it. Two types of inhibitors. Competitive inhibitors, that's lion bubblegum. All right? It goes into the active site, and then the other substrate can't get in there because the lion's mouth is full of bubblegum, can't bind to the gazelle. Cool? There are also non-competitive inhibitors, okay? As I said before, all inhibitors affect the enzyme's, the rate of enzyme catalyzed reactions, the enzyme's activity by affecting the enzyme's active site, okay? Competitive inhibitors directly bind the active site and block it, whereas non-competitive inhibitors, they bind somewhere else on the enzyme and they change the active site's shape, okay? Cool? So let's say the lion's getting bit by a big bug. The lion's got a big bug on its butt, and it makes it frown. And that's changed the shape of its active site. And a frowning lion cannot eat gazelles, which is, what my father always told me. He had, he liked his drink, this man. And he'd say to me, Jonathan, a frowning lion cannot eat gazelles. And I'd say, yes, Father, I love you, Father. And he'd say, now, out, chop the wood for the fire, for tonight we feed on frowning lion. Is this working for you? All right, whatever. Let's keep moving forward. Oh, man. Now I'm thinking about my dad. That's a bad thing. That's bad. Okay, regulation of enzyme activity. We cool? Let's talk about this. This is funner. This is more fun to think about. Enzymes are proteins. They're encoded by genes. So, yeah, you can change an existing enzyme's shape, change the shape of its active site, or change the availability of the active site by binding it with a competitive inhibitor. Or, if you don't ever want the enzyme to be present, just shut the genes off that make the enzyme. Okay? If you never transcribe and translate the genes that encode the enzyme, you'll never have the enzyme, and you never have to worry about regulating its function. And then if you need the enzyme, remember all the cells have the same genetic code in them. So if a cell's changing type, or if a cell's developing and at this stage of development it needs to change what enzymes are present, just activate transcription of the enzyme. Start making the enzyme, okay? So regulating the enzyme gene is a very long-term effect. Because remember, once you've made the enzyme, it's going to do its thing until it eventually breaks down. So it can do hundreds of rounds of it can catalyze hundreds of rounds of reactions. All right? Cool. Or you can regulate the activity directly through inhibitors. Questions about this slide? I hope I laid the groundwork effectively for just how important enzyme regulation is in determining cell type, cell function, cell activity. It's huge, okay? I can't overstate how important it is, and it's very easy to overlook how important enzymes are, okay? Let me just tangent it for a second here. There are a lot of things that differ between your different cells, right, that aren't just protein. But the only thing that you directly make is the protein, right, by transcription and translation from the DNA. But all those other chemicals that are present at different levels in all the different cells, those are there because of what metabolic reactions happened. And all of those metabolic reactions are determined by enzyme activities. Okay, I'll get off it. That's good. Let's talk about inhibition. Feedback inhibition. Feedback inhibition is not a phrase that is specific to enzyme activity. In feedback inhibition is a general term for I am a reaction and I make a product. The product inhibits the reaction. Okay? I am the thing that converts closed staplers into open staplers. But I keep track of how many open staplers there are. And once there are enough open staplers, the open staplers actually inhibit my activity. And now I don't convert staplers anymore. All right? That's feedback inhibition. A textbook example that they love is a thermostat. All right? Which I don't like it too much, but, you know, if they put it in a textbook, it must be good, right? So you turn up the thermostat, boom. The thermostat makes heat, all right? The heat is the product of the reaction, and once heat levels get high enough, that inhibits the reaction making more heat. See what I'm saying? In feedback inhibition, the product of the reaction feeds back and stops the reaction from proceeding. So right here, you have an enzyme catalyzed reaction. And this is the first slide where I really officially introduce, there's usually multiple enzymes in a metabolic pathway. All right? So a molecule will go from like molecule A to molecule B to molecule C to molecule D to molecule E to molecule F. And F's the final product. But each step along that pathway, I do it like this, I think, yeah. Each step along that pathway is catalyzed by a different enzyme. All right? I'll give you an example of it later. It is also, everything's important. I really try to remove all the chaff from these summer courses. So every slide has something key that I'm trying to convey to you. All right, let's focus on this one, which is feedback inhibition. But don't forget there's a multi-step pathway. Question? Oh, the feedback inhibition? Yes. Because now you have enough of the product. And that's the idea. That's the idea of feedback inhibition. It's very rare that I can say yes without a bunch of caveats. But yes, with feedback inhibition, you have enough of the product and you don't want to make more of it. Because in the metabolism of a cell, this starting molecule A is probably used to make like 20 different things. And you don't want to be wasting it making stars when you also have to make hexagons starting with it, right? So this is and the system needs to be simple because it's chaotic inside a cell. You see what I mean? Like you don't have a multicellular supervisor being like, stop, we have enough tires, stop making tires. You know, the tire has to actually wrap around the person who makes tires to make them stop. And then when you really need tires, you'll pull the tire off of that person and allow them to make more tires. Which honestly might be a pretty good analogy. I don't know. Let's see if it continues to work. We're testing it out in this summer course. So check this out. Multi-step reaction, multi-step metabolic pathway. Starting molecule A, Enzyme 1 facilitates or catalyzes reaction 1, which produces molecule B. Molecule B, enzyme 2, facilitates reaction 2, which converts molecule B into molecule C. Enzyme 3 catalyzes the reaction where molecule C goes into molecule D, the end product of this multi-step metabolic pathway. So far, so good? Product D binds to enzyme 1. So if there's enough product D, that will inactivate all of the enzyme 1. And that will stop this pathway from going forward so that A can be used for other things, or we're just not wasting energy because ATP might be involved with some of these enzymes, right? So you have enough of it. So you need to stop it, right? Cool. All right. And eventually, as the cell is using up product D, it will pull this off and use it up, freeing up the enzyme to make more of product D. Okay? We cool so far? Awesome. This process is also not permanent, right? So you think about all of these products floating around in the system, and whenever they hit an enzyme, they go like, oh, I'm going to bind here for a while and stop that enzyme from working. Then they go back into solution. But if there's a ton of them, then they're constantly binding the enzyme. That means that as you reduce concentrations of product, the time where the inhibition is occurring becomes less, because there's more time between a product molecule coming by and happening to bind and inhibit. Kind of makes sense? So it makes for a really beautiful system where the reaction gets ramped up and ramped down because there's a bunch of enzymes floating around here. It's pretty cool. Feedback inhibition's pretty cool. That's a cool guy. That's what cool guys do. Yep. All right. Here we go. Now it looks more complicated, but it's exactly the same thing. There's names of molecules, and you don't have to worry what those molecules are. But this is just classic, you know. You start with it simple. You make it look more complicated without adding anything more to it. And this sort of stretches your brain a little bit in understanding. Let's take a look here. Enzyme 1, enzyme 2, 3, 4, and 5. There are 5 enzymes in this pathway. Initial molecule's up here. It's a molecule called threonine. What's threonine? It's 1 more than 2-anine and 1 less than 4-anine. Don't worry about it. Thank you. All right. It binds to the active site of enzyme 1. Boom. Makes molecule an intermediate A, which binds to enzyme 2. Boom, 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 boom. In product, isoleucine. Isoleucine, also, please don't worry about it. Isoleucine is the product of this multi-step enzyme-catalyzed metabolic pathway. Cool? Check it out. This end product binds to enzyme 1, but not to the active site. This is not competitive inhibition, and feedback inhibition is usually not competitive inhibition because the product is inherently different from the substrate, right? So there won't be any affinity between the product and the active site. There kind of can't be. So feedback inhibition is usually non-competitive inhibition. Check this out. Isoleucine, the end product, it binds to a separate site on the enzyme, right here. And when it's bound, the active site changes its shape. Frowning lion. All right? Gets bit on the butt, frowns. All right? This site that is not the active site and exists entirely for regulating enzyme activity is the allosteric site. Okay? This here is non-competitive inhibition because the inhibitor, which is right now isoleucine, is operating as an inhibitor, language logical, cool? It does not compete for the active site with substrate. So it's not competitive for the active site. It's non-competitive. The site it binds to is the allosteric site. And this type of inhibition is also called allosteric inhibition. Cool? Because the site that the inhibitor binds to is the allosteric site. Questions? Good. Now, as the product gets used up in the cell, all right, there will be less as isoleucine gets used up by the cell, there will be less isoleucine available to bind allosteric sites. Remember that this isn't permanent, it sort of bonds and then falls off. Okay? That means the enzyme can become more active. This is feedback inhibition. Any questions about feedback inhibition? Okay. Overall, nice slide here that shows both types of, well, this actually shows allosteric inhibition only, but also introduces the concept of allosteric activation, which just complicates the matter, of course, because that's what happens to matters. They get complicated. Yes? No, because I hate to give a no, because that's great thinking, is what I should have said. But the cofactor, remember, binds to the active site and is actually necessary for the function of the active site. An allosteric activator, it isn't necessary for the active site to function, but when it binds to the enzyme, it changes the shape to a functioning shape. And it's really splitting hairs to, say that's different, but it is not functioning as a cofactor there. Awesome question. You can have 2 points, of course. Good. Competitive, non-competitive inhibition. So far, I think we're all feeling pretty good about it. Okay. I'm gonna, kind of keep my pace up a little bit and just slow me down if you have any questions. Okay, check it out. Inhibitor, allosteric site, changes the shape of the active site. That active site can't bind substrate, the enzyme doesn't work. Allosteric inhibition. There are also examples of allosteric activation. Sometimes you only want the enzyme to be active when something that is not the substrate is also present. You see what I mean? It's kind of weird. But sometimes you'll use the presence of one molecule to indicate that, oh, I need to break down something else. I need to become an active enzyme. But I'm not actually breaking down this indicator molecule, okay? So this would be an example of allosteric activation, where a molecule binds the allosteric site and makes it open up and become active. Do you have a question? Oh, sorry, I thought I saw a question up. No problem. Okay, so the inhibitor is the thing that inhibits the enzyme. An activator activates an enzyme. Nice language, right? Makes the active site work. Cool? Please, yes. Of a competitive inhibitor? Absolutely. So, no stress. I got it right here on the next slide. Some of the best competitive inhibitor examples are like synthesized drugs, okay? Like medications. Not like, whoa, drugs, but like, drugs. You know what I mean? If enzymes aren't regulated effectively in your body, you can end up with a bunch of metabolic pathways that don't work right. And that's no good. Accumulation of certain things that you don't need. You use up a bunch of precursor molecules that could have been used for other things. If you can develop a molecule that is similar to the substrate molecule, but won't be broken down by the substrate molecule, won't be broken down by the enzyme, then the active site becomes filled, okay? And then the enzyme can no longer function, all right? I'm so embarrassed. I usually have locked and loaded in my head an example, and I just don't. But sometimes you'll see this is actually really, okay, this is interesting to me, so you have to listen to it. All right? It's a marketing strategy. In advertising, if you can make the person you're advertising to feel like they were educated by the advertisement, that can get them more likely to purchase the product. It's like one of those principles of advertising. I wanted to be in advertising when I was a high school student, but I don't know. I wasn't really encouraged down that pathway. And so now I'm here. And I'm trying to encourage all of you. I'm advertising interest in science, I guess, because everything's marketing, right? Anyways, here's the thing. Keep your eye out on, like, if you ever have to watch cable for some reason, keep your eye on the medication advertisements, because they kind of go two routes, right? It's like, here's what your life could be like if you took our medication. This will also kill you, it's terrible, right? That style. Or you find the educational track. And in the educational track, they'll say, our drug blocks a trigger that tells your body to have eczema. And then you'll see an image and it's like, oh, it's showing binding to a receptor protein on a cell. Like, it's trying to use plain language to talk to you about a chemical that blocks the activity of a receptor. You'll see those same advertisements for drugs that say, this stops your body from naturally breaking down bleh. All right? And that's pretty darn effective in keeping the levels of bleh high in your body. So you don't have to take a supplement for bleh. You just stop your body from breaking bleh down. Anyway, keep an eye out for that style of marketing. It only comes up every once in a while, but it's interesting when it does. You know, it's way more common to be like, hey, drive fast car, drink Coca-Cola. But it's much less common to be like, Coca-Cola can make you happier, and being happy is good. Something like that. It's educational. I drank a big Diet Coke last night at the movies. I saw that new Ari Aster A24 movie. You know, it's pretty terrible. It's a good movie, but man, I was sad. It takes place in 2020 in a small town in the midst of COVID. It's the most depressing thing I've seen. But if you've seen Hereditary, what are you surprised by, John? You know what Ari Aster is going to do to you. Yeah, okay, cool. There's John's movie corner update. Then I came home and watched Spaceballs with my partner. And we felt a lot better after that. All right. Yeah, they did the whole thing. It's like this penis, it's funny. Yeah, you got to alternate. Anyway, yeah, you do down, up, and keep your brain freaking out. Anyways, inhibition, enzyme inhibition. Let's take a look at this figure right here. This is a cool figure, one that I'm not going to test you on, but I encourage you to read in your textbook if you're interested in this at all, because it really brings all the concepts sort of back together and makes you really think about how is inhibition actually working, okay? Competitive and non-competitive inhibitors function differently inside the cell. So if you're taking biochem or genetics, they'll talk to you probably about how, oh, the body is going to be using competitive inhibitors here and non-competitive inhibitors here because of how tightly it needs to regulate the reactions, how important it is that the enzyme stops functioning, basically. So check this out. This shows not a time course on the x-axis, but rather an increase in substrate concentration on the x-axis. And on the y-axis, it shows you a rate of reaction. Now, enzymes, substrates, they are moving around randomly in the system. I talked to you about this at Wednesday's lab, all right? They're moving around via Bayesian motion, which is just a way of saying that they're randomly moving around in all directions. Sometimes they hit each other, and then boop, 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 the product gets produced, okay? So if there's more substrate floating around, it's more likely that the enzyme will encounter a substrate molecule. Therefore, increasing substrate concentration should increase enzyme reaction rate, okay? the rate of the reaction catalyzed by the enzyme. Is that logical enough for everybody? There's going to be a flat line though, because at a certain point, every active site of every enzyme is occupied by a molecule of substrate. And that's the limiting factor. So in this figure here, the concentration of enzymes is always the same. At a certain point, you keep dumping substrate in, but each enzyme is working as fast as it can. It's constantly encountering substrate. Does that make sense? So you have a plateau of the rate. Now, competitive inhibitors compete for the active site. That means that if you keep dumping in enough substrate, you can make there be more substrate than competitive inhibitors. And so you can get the reaction rate to reach about the same level because these are the inhibitors. All right, there's only a number of these, but there's so many substrate molecules that they're going to be more likely to encounter. And every once in a while this happens, but then one of these goes right back in there. Remember, this is all transitory. Transitory, I think that's right. Non-competitive inhibitors, they bind the allosteric site. So you can actually cap the rate of the reaction more effectively because it doesn't matter how much substrate you add in. A lot of the enzymes just don't have active sites that are shaped right. But remember, okay, so if that's how it works, why is there any enzyme catalyzed activity? Because the allosteric inhibition is also not permanent. It's designed to not be permanent. Because enzymes are big, they take a lot of energy to make, and you don't want to be breaking enzymes down when you don't want them to be active, and then having to generate whole new enzymes when you do need the enzymes to be active. Woefully inefficient. Okay, kind of makes sense. Like I said, if it didn't fully make sense, it's kind of nice. It brings a lot of stuff back together, but I'm not planning on testing you on this aspect of enzyme inhibition. Okay, we're almost at the cool. Let's just rock to the end of this. We have about 8 minutes left, and we only have like effectively 3 slides left. Okay, Roundup. All right, we're afraid of it. It's bad. Roundup Ready crops. Not sure if it's still a big buzzword, buzz phrase for students here. Not so much. Used to be a big thing. A lot of people in the biological sciences were afraid of Roundup Ready crops and what they meant and all these things. Okay, cool. So check this out. Affecting enzyme rates is hugely important. I think I have emphasized that to you enough, right? If I prove it's fine. Yeah, it's fine. There are lots of beneficial drugs that work as enzyme inhibitors. If your body's not regulating its enzyme activity correctly, boom. All right? Prevent reuptake of something, prevent breakdown of something, okay? But enzymes are so important that inhibiting enzyme activity also can kill things. Pesticides, herbicides, chemical warfare, nerve agents, sarin gas, all right? Sarin inhibits this enzyme cholinesterase, all right? Which, if I'm not mistaken, it adds ester groups to cholines or something. I always forget. I used to know, all right? Or Roundup, okay? Glyphosate is the name of the actual chemical that's used. It works incredibly well, okay, at killing broadleaf plants. You spray it on a plant, and it inhibits this enzyme here that's essential in generating 3 different amino acids. And if the organism can't make the amino acids, it can't grow, it can't live, it can't make enzymes, it can't make any proteins. All right? So that's how glyphosate sort of works, is it inhibits that enzyme. And that's enough to kill the plant entirely. Now, a Roundup Ready plant, what they have is they've been genetically engineered to express, and this got cut off a little, sorry. They've been genetically engineered to express a bacterial form of the enzyme. And this bacterial form of the enzyme is not inhibited by glyphosate. So you genetically engineer a plant and you make it Roundup ready, and no matter how much glyphosate you spray on it, it keeps making the amino acids. So if you're doing agriculture and you want to keep your big field of corn free of weeds, then you just make Roundup ready corn, and then you just spray the field with Roundup, and that kills everything else. There you go. Problem solved. All right. Now, Roundup ready crops, big, a lot of controversy around them, a lot of conflicting data about if Roundup ready crops can actually hurt people who are eating the Roundup ready crops. There are other plants that are genetically engineered to produce their own pesticides. All right. BT corn is an example of that, where it makes a bacterial enzyme that breaks down the stomach lining of insects that eat the plant. That's different, but it's pretty cool. And then they've got what they call like triple stack corn, where it makes its own pesticide and it's Roundup ready and it's resistant to another herbicide. And that corn, you can just dump poisons on it all day. Bugs try to land on it, their intestines dissolve, but then we eat the corn. Intuitively concerning. The data to actually support that Roundup ready crops hurt humans or animals, clouded by on the one hand, sloppy science and very vocal science, and on the other hand, very well-funded science. It's a serious issue, you know? And now, seems like it may have faded into the back of our minds. Ooh. All right, anyway. Compartmentalization of enzyme activity. Why do the organelles do different things? Because they have different enzymes. I'm going to oversimplify the hell out of it and just sort of leave it at that, all right? Mitochondria have this super folded inner membrane that we've talked about already, and it is just studded with these enzymes that are involved in cellular respiration. Remember how I talked to you about how membrane proteins can be enzymes? Remember that? Cool. Cool. I love it. Great. Laundry detergent plus enzymes. All right. Mark Wilson, Patty Searing. They were partners. Patty Searing's passed. It's very sad. They ran the heat lab on campus. Okay, I believe the heat lab still functions as of like a year or 2 years ago. They're involved in looking at acidophiles. They go to like Lassen National Park and try to figure out what's living inside of extreme environments. Now, how do you get funding for that? Because who cares, right? I, in my heart of hearts, have no interest in reading a paper that tells me which archaeans and bacteria live in a volcanic hot spring. Are you going to read that? I'd like to think, I think that, maybe when I was younger I would have, but I worked in the heat lab. I never read any of those papers, But hey, you find a way to make it into something that has something profitable possibly, then you got funding for your research. Now people are interested, okay? So enzymes and detergents, great. Break down proteins, break down starches, break down, carbohydrates. Get your clothes cleaner, okay? You already know detergents are pretty good at breaking down hydrophobic substances, all right? How about some enzymes in there as well that break down things within the hydrophobic substance, all right? Like a protease, that breaks down a protein. So these come from bacterial origin usually. You get enzyme detergents all the time, all right? And usually they're isolated from extremophilic organisms, usually extremophilic bacteria, okay? Because the laundry machine is an extreme environment, right? So the enzymes need to be stable at basic pHs and at high temperatures, all right? This is where Patty Searing made her industry money before she came to teach here at then HSU is she developed like Tide. She helped Tide get, these good enzymes. And I really honestly keep the slide up because I feel I miss Patty. She was great. She really encouraged me when I was an undergraduate, and this slide was up because of her. Anyways, whatever. It's fine. It's fine. The last thing, here's the very last thing. I'm gonna go 1 minute into your lunch. I'm very sorry, but I'd like to finish up the slide. Last thing, cofactors. Cofactors, they are not proteins. They are required for the enzyme to function, or at least are required for the enzyme to function at the correct rate, the max rate, okay? Sometimes you'll have a cofactor that is an organic molecule, okay? So we've already talked about cofactors that are non-organic, like a magnesium ion or an iron ion. They're not carbon-based molecules. They're a single atom, right? We cool with that concept? Sometimes you got organic cofactors, where there's a big carbon skeleton with some functional groups sticking off of it. That's technically called a coenzyme. It's still a cofactor, but it's more specifically a coenzyme. Coenzymes include vitamins. So why do we have to take vitamins? Because they are necessary for enzyme functions, all right? Not all vitamins are this. Now, specifically, a vitamin is a coenzyme that you do not make in your own body. So you have to supplement it. And someone will always say, because you're critical thinkers, someone will usually say, what about vitamin D? All right? And the thing here is that that's not really a vitamin because we can make it internally, but supplementing it is usually necessary to reach, the levels that you like. So sometimes you call things vitamins when they're not technically purely vitamins. Any questions about that? Cool. We'll come back to respiration tomorrow. Then we'll talk photosynthesis on Wednesday. And then we'll have lecture exam on Thursday, which will cover material up to the end of this PowerPoint. We are on slide 38 of 96 right now. If we get all the way through the slides by the end of the day Wednesday, I will use the remainder of our lecture time to answer any study guide questions that people have. So start working on that study guide now and bring your incomplete one Wednesday, and I'll help explain things. Thank you all so much. I'm sorry, I stole 5 minutes of your lunch. That was really rude of me. But you're free until 1 o'clock, and we're going to do pigments. I'm going to stop this recording.